Good morning, everyone. I hope everyone's doing well, and thanks for tuning in today and listening to my talk on mechanisms of inflammation in lipid-laden macrophages after spinal cord injury. So, you know, after spinal cord injury, there's kind of a cascade of cellular events that occur in response to mechanical damage to the tissue. The first stage being mostly mediated by resident glia cells, such as microglia and astrocytes. And they kind of perform the first wave of inflammatory signaling. And at that point, we then have the infiltrating immune cells that come in, which includes, but is not limited to, monocytes and macrophages. And they, in concert with the resident microglia and astrocytes, perform a number of functions at the injury site, including removal of debris, promoting angiogenesis and revascularization. They're also involved in the kind of scarring response in isolating the lesion site, and at the end, hopefully helping facilitate tissue regeneration across the lesion site. However, there is a lot of controversy and a lot still unknown about the role that these immune cells, particularly macrophages, play in the response to injury. And we've seen reports of beneficial as well as detrimental responses. And so this has been a major interest in our lab's research, is to really understand what role these macrophages are playing in response to injury, and if they're either helping or contributing to the cellular pathology. So to get a really unbiased look at these cells and what they're doing at the injury site, we utilized a special technique, ribotag technique, which enabled us to isolate blood-borne macrophages specifically, and isolate their RNA at the injury site. So what we did is we compared the transcriptome of macrophages taken from the 3-day post-injury site and the 7-day post-injury site. And what we found was that when we compared their transcriptome, the processes that were highly upregulated at 7 days were, to our surprise, largely lipid, metabolic, and catabolic associated processes. And again, this was surprising to us, and even more so when we took the transcriptome of these cells and compared them to publicly available RNA sequencing data sets. So the transcriptome of these 7-day post-injury macrophages at the injury site actually most closely matched those of foam cells from atherosclerosis. And so, again, this was not expected by any means, but was very interesting to us. So, what are foam cells? Maybe your next question. So, as I mentioned, they are largely studied or were originally identified in the context of atherosclerosis. So, similar to spinal cord injury, there's inflammatory signaling that cascade that occurs. There's a deposition of lipid debris in the artery wall, in this case, instead of the lesion site. And so macrophages are recruited to this lipid deposit. And essentially, as we know, macrophages are the professional phagocytes of the body. So they are recruited to this lipid deposit and begin taking up this lipid debris, both through non-receptor mediated mechanisms as well as receptor mediated mechanisms. So what you can, so once these lipids are tested, there's a variety of cellular signaling events that occur, and they usually are incorporated into the lysosomal processing center of the cell. But basically, in the case of the macrophages or the foam cells, their ability to process and export these lipids effectively is basically overwhelmed. And they start to form these lipid droplets in the cytosol, which is how they get their name. Foam cells is from the presence of these lipid droplets. So in addition, the presence of free cholesterol in the cell from this inability to clear and export these lipids can basically ignite the induction of some pro-inflammatory signaling, such as the NF-kappa B pathway, as well as the accumulation of cholesterol crystals, which can activate the inflammasome and can also induce lysosomal dysfunction. So, in spinal cord injury, the same principle applies. And in this case, instead of cholesterol in the artery, we instead have a large amount of cellular and lipid debris from the damaged cells. That's basically macrophages are coming in, same way, and attempting to clear this debris, but essentially become overwhelmed with the amount of this debris and become filled with these lipid droplets. So other labs have also been interested in the presence of these foam cells after spinal cord injury. Another lab, notably Yiren's lab at Florida State. And so this is a staining of the injury site over time. And this is oil red O, which is a neutral lipid dye. And basically, you can see around, we have our uninjured cord up here, around 3 days post-injury, this is when macrophages are entering the cord, but haven't really started to, they're just starting to ingest and process the tissue. So by 1 week, we start to see the presence of lipid droplets, as you can see by the oil red O stain. By 2 weeks, the signal has really amplified, and by 4 weeks, we see the development of this really lipid-rich lesion core, and you can see the formation of these cells that are basically engorged with lipid droplets and have kind of condensed into this lesion core. And so when most people wonder what's the source of the lipids at their injury site, obviously their first thought is myelin, which we know is essential to nervous, the function of the nervous system. So the kind of status quo for testing these cells in vitro is to treat macrophages with purified myelin. And so this is a graph from the same paper where they treated macrophages in vitro with purified myelin and found an increase in a number of pro-inflammatory cytokines. And so this kind of set the stage for, potentially implicating these foam cells in propagating, pathological inflammation at the injury site. And definitely put our own just findings with the 7-day post-injury macrophages kind of in perspective. So the overall, I guess, the big picture of my project is to basically get a better understanding of these foam cells and how they're contributing to the cellular pathology after spinal cord injury. And so first on my agenda will be to discuss the role of myelin specifically. I kind of just mentioned this in the previous slide, that there is obviously this is an important source of lipid at the injury site. And so this has kind of been the lipid most people have focused on with foam cells, but it has never really been directly tested. And so this was something we really wanted to take a more careful look at. Additionally, I was interested in the downstream consequences. After lipid uptake and we start to get the accumulation of these lipid droplets, what are the intrinsic cellular changes occurring as a result of this? And specifically, the metabolic changes that accompany this. We know that lipids are very difficult to digest and export, and so this puts quite a bit of stress on the cells. So I was interested in really taking a more careful look at the metabolic profile of these cells. And lastly, I also was interested in potentially the downstream inflammatory signaling events that may be occurring as a result of this lipid accumulation and metabolic dysfunction. There's some precedent, as I showed in the work from Yuren's lab, where we saw this increase in pro-inflammatory cytokine signaling. This has also been shown in the context of heart disease and atherosclerosis. But I wanted to, I guess, get a clearer picture of what exactly they're contributing as far as their inflammatory profile, and whether or not this is helping or harming the injury site. So to first address this problem of myelin and what exactly it's doing to induce foam cell formation at the injury site, we actually utilize this very special mouse model that has myelin basic protein completely knocked out. So as a result of this, they're essentially depleted of all central nervous system myelin. And due to the impaired axonal conductance due to the absence of myelin, they exhibit this tremor, which gave them the name shiverer mice. And so to address this gap of knowledge of what truly is the contribution of myelin to the formation of foam cells after injury, we wanted to use, again, this mouse model that, as you can see here, this is a Luxel blue staining, and this is the wild type mouse, which you can see the white matter is robustly labeled with the Luxel fast blue, and in the shiverer mouse, completely absent. Additionally, this is just an immunohistochemical stain for MBP. You can see the wild type, the longitudinal white matter tracks labeled strongly with this MBP protein, and again, this is completely absent in the shiverer mice. So to directly test the role of myelin in foam cell formation, we wanted to take this existing in vitro scheme that was utilized by a number of labs, which basically meant they treated naive macrophages with purified myelin to induce foam cells. Instead, what we wanted to do is take the same source of macrophages, take bone marrow-derived macrophages, culture them to maturity, and then instead of purifying myelin from the spinal cord, we wanted to take the entire spinal cord homogenate. And so we took cord from these MBP het mice, which have a completely normal amount of myelin, and compare them to cells that are treated with MBP knockout homogenate, which again is depleted of myelin, but has all other sources of lipids, such as general cellular debris. So we have this homogenate in our inner media, and we treat our cells with this mixture for 24 hours. And then we use this to basically produce our foamy macrophages, and this is what we can use for downstream analysis. And so what you can see here is these are the cells that I treated with MBP het homogenate, which is shown in this top group of this top panel, and the cells treated with the myelin deficient MBP knockout homogenate at the bottom. And what you can see is there is, when you look at each, this is also increasing percentages of the homogenate. So as you can see, with increasing percentages of homogenate, we're getting an increase in lipid content, which is here labeled in green by another neutral lipid dye known as Nile red. And so as we knew and expected, that these cells treated with the regular spinal cord homogenate are going to basically start forming these lipid droplets and become basically filled with these lipid droplets. However, when you also treat them with MBP knockout homogenate, which again has absolutely no myelin in it, you're still getting the formation of lipid droplets. It is decreased relative to the same cells treated with homogenate that contains myelin, but this really kind of dramatically showed us, though, that myelin is not the only source of lipids that's driving foam cell formation. You can see here, this is just a quantification of the lipid droplet fluorescence. We have our cells treated with the MBP knockout, het homogenate in black here, and at the same percentage, cells treated with MBP knockout homogenate, again, it's about a 40 to 50% decrease, which is just, again, underscoring that we really were missing a large part of this picture by treating with purified myelin, and that these other sources of cellular debris, just general, injured, damaged, dying cells, are also an important source of these. And so we also obviously wanted to test this in vivo and see if the same phenomenon is observed. So we took the MPP het and knockout mice and subjected them to a contusive spinal cord injury. We then stained for neutral lipids using oil redo, a neutral lipid stain, and CD11B to measure or to label myeloid cells at the injured site. And so we have our NDP het animals here on the left. This top panel, you can see both the lipids as well as the myeloid cells. And so as we expect, we get, lipid droplet filled macrophages in the MBP het mice, which basically contain myelin and are similar to a wild type injury. In the knockout mice, which again do not contain any myelin, we do get a decrease in the oil redo signal. However, as you can see, foam cells still are forming. And so this is just basically driving the point home that myelin is indeed important for foam cell formation, but it's really only accounting for about 40 to 50% of the lipids that are constituting the foam cells at the injury site. And this is quantification for the oil redo signal. Again, at 7 days post-injury, we have a decrease in coverage, as well as a decrease in the oil redo fluorescence intensity. And at later time points, at 28 days post-injury, you can see this oil redo signal has kind of consolidated to this lipid-rich lesion core, similar to what we saw in Uren's histology. And we're again seeing the robust formation of these foam cells in both the NDP het as well as the knockout mice shown here. So again, we get about a 50% decrease in our oil redo area coverage and our oil redo area fluorescence intensity, but that foam cells are still forming in the absence of myelin. So to also understand what's the consequence of reducing foam cell formation at the injury site, in this case by removing myelin, we also did a stain for GFAP, as well as nu-N, which labels neuronal cell bodies. And so the way we measure the lesion size, the spinal cord injury lesion size, is to measure the GFAP negative lesion area, which I have outlined here in aqua. And so as a result of this reduced foam cell formation, we do get a decrease in the GFAP negative lesion size, which is also correlated with increased neural tissue sparing as a result of obviously having less of a GFAP negative lesion area. We're going to have more spared neural tissue and more neuronal bodies in the proximity of the injury site. And so again, I found the same thing at 28 days post-injury. We're seeing this decrease in the GFAP negative lesion size in these MVP het mice that contain myelin, and that this is decreased in the MVP knockout mice, which do not contain myelin and have less foam cells. So as interesting as this was, we all know that histology can be sometimes difficult to quantify exactly what's going on, although it does give you a good obviously visual understanding of the dynamics of the injury site, but we still wanted a more quantitative understanding of the cell types that are present at the injury site as a result of macrophage lipid accumulation. So we wanted to take a more careful analysis of the different immune cell populations that are infiltrating the immune site, and again, compare this in our MVP het mice, which have the normal amount of foam cells and normal amount of myelin debris, versus the MVP knockout mice, which have reduced foam cell formation. So this is just our gating strategy that we use to identify the standard immune cell populations. So we have here, we identify our singlets, and then we get our viable CD45 cells. We use Ly6G to gate out our neutrophils. And then of this Ly6G negative population, we get our macrophages and CD45 low microglia, and then our CD11B negative lymphocytes, which can be further subdivided into our B cells and T cells. And this is important for our application because obviously in our histology, CD11B is going to label both macrophages and microglia. From what we know, based on some bone marrow chimera studies, we do know most of the CD11B positive cells in the center of the lesion are indeed macrophages, but it was important to us to really carefully separate these two populations and understand their contribution to basically their lipid content at these time points. So this is just our controls for ensuring our gating is properly done. Here we have an example of the injury site, flow plot, and then we always would take a rostral and caudal section of the injury site. So this is the injury site taken, shown here. And then we would take also as a control this rostral and caudal section, which we know does not contain many macrophages. And so as you can see here, most of these cells are falling under our microglia gate. Conversely, we would also take the spleen, which obviously shouldn't contain any microglia and just macrophages. And we again see that this, our gating strategy is good, and we're good to go. So this was also a very surprising finding. So what we saw was we have our MVP het injury site, flow plot seen here on the left, and our MVP knockout injury site seen here on the right. And obviously you can see in this macrophage population, the CD45 high, CD11B positive population, there's a large decrease in the number of these cells at the injury site. So this is the quantification for that. Here, obviously, you can see this large decrease in macrophage number in the MVP knockout cells. We did not see any difference in microglia number. Additionally, it also looks like these myelin deficient mice kind of also exhibit a global reduction in leukocyte infiltration, and that included a decrease in overall lymphocyte number, and specifically a decrease in the T cell population. So now that we have these immune cell populations separated out and quantified, we also wanted to understand what their individual lipid content of these different populations were. So we also used another neutral lipid dye known as Bodepi. And basically you can use this as another readout for lipid content. Here we just have a validation study that I did using my in vitro assay. And so I have here my media-treated macrophages, which shouldn't really contain any lipid droplets. And over here we have my macrophages treated with the 1% homogenate. So we have this conversion to the foam cell phenotype. And as you can see here, the Bodepi signal is represented here on the y-axis. And you can see this pronounced shift upwards to this Bodepi positive population. This is the Bodepi fluorescence histogram. Again, you can see this large increase in Bodepi fluorescence from our media-treated macrophages to our foamy macrophages, just confirming that our dye is indeed labeling what we wanted to label. So when we looked at the number of macrophages that were Bodepi positive or Bodepi high at the injury site, as is no surprise, there was a huge decrease in the MVP knockout mice. However, as we saw before, there was also just in general a large decrease in macrophage number. So what we did is we also normalized this Bodepi positive macrophage population. We normalized it to overall macrophage number. So here we have it expressed as a percentage of total macrophages. And here this decrease is kind of more on the order of what we saw in our histology. And so again, there's a decrease in the number of Bodepi positive macrophages at the injury site, but it is again not accounting for the entirety of the foam cell formation. And so all these results, they make sense in light of a lot of work that has been done studying myelin as a chemotactic cue. So here, this is a study from a paper done by, again, the same lab at Florida State, Uren's lab. And basically here they injected myelin into an injured spinal cord and measured the immune response to that myelin injection. And when they knocked out complement receptor 3, which is known to be an important receptor for myelin, basically you see a loss of this immune cell response. We have our EVO1 cells, which also labels myeloid cells. And you can see in the wild type mice that injection of the myelin causes this robust recruitment of immune cells. But when you knock out this receptor for myelin, that you do not see the same response. So this is all again consistent with myelin itself acting as a chemotactic cue, and would maybe an important piece of evidence in understanding our flow data from the MVP knockout mice. So in conclusion, for this first part of my talk, myelin is, again, a source of lipids that is driving foam cell formation, but it's not the only source, and it is not even the most important source, and that other sources of lipid debris, notably cellular debris from damaged and dying cells, is responsible for the rest of this response. And in vivo, we also saw that decreased oil-redo positive macrophages also correlated with a decrease in GFAP negative lesion size in the MVP mutants. Again, all of this is just underscoring our the importance of our modified in vitro assay, in which we use the entire spinal cord homogenate and not the purified myelin in order to induce our foam cell formation. And lastly, our flow data confirmed a lot of our, histology, but also provided some more information on myelin and its role as acting as a chemotactic cue for immune cell infiltration after injury. So next on my agenda was to understand again some of the intrinsic changes that are occurring as a result of this lipid accumulation, and specifically some of the metabolic changes that may be occurring in foamy macrophages after spinal cord injury. So to address this, we utilized a really cool tool known as the Seahorse mitochondrial stress assay. And basically what this machine allows you to do is in real time to measure the oxygen consumption rate of your cells. So basically you can take cells in vitro, in our case macrophages, treat them or not treat them with homogenous. We have our non-foamy macrophages, and then we insert them into this machine, and it can basically start reading how quickly they're consuming oxygen in real time. And this gives you an idea of their oxidative state. And so when you first insert the cells into the machine, they take a baseline reading of their respiration. And this is without any other compounds added. This is just the cells, in this case, our foamy cells or our non-foamy cells, just in regular media, and just to get a baseline of their metabolic state. Next, there's little injection sensors in the machine that inserts oligomycin into each of the wells. And oligomycin is a ATP synthase inhibitor. And so this will basically reduce the flow of electrons through the electron transport chain. And what you'll get here is a decrease in oxygen consumption rate as a result of ATP synthase inhibition. And so they correlate this to ATP production because obviously ATP synthase is responsible for ATP production. However, I think it's important to note that you're not directly measuring actual ATP content. You're just measuring oxygen consumption related to ATP synthase activity, which is an important distinction. Additionally, the remaining oxygen consumption that occurs after this oligomycin injection is known as the proton leak, because at this point, any remaining oxygen consumption is going to be due to leakiness across the mitochondrial membrane, which is driving still the proton motive force, because at this point we've inhibited oligomycin, which is normally responsible for regulating that response. And so proton leak is also important for understanding kind of the general health and the mitochondrial integrity of the mitochondrial membrane and its function. So then next we inject FCCP, which is a uncoupler. And basically this takes the brakes off the system, so to speak. And so when you completely uncouple the gradient across the mitochondrial membrane, this enables you to basically drive the cell to its maximum respiration state, because what was normally basically inhibiting, or not inhibiting, but regulating and determining the rate of oxygen consumption is completely uncoupled now. And so again, you can bring the cells to their theoretical maximum. Lastly, you inject this rotenone and antimyosin mixture, and this basically shuts down the remaining parts of the electron transport chain. And at this point, the entire chain is completely non-functional, and so you get a complete shutdown of oxidative phosphorylation in these cells. And so what you have here is this remaining oxygen consumption is due to non-mitochondrial processes, and so this is a value that you subtract from all of your other measurements. And so another important measurement here that you can get from your maximum respiration is known as your spare capacity, which is basically the amount of maximum respiration that you have after you account for your basal respiration. So this is also, in addition to proton leak, and another important readout of mitochondrial function, and the spare capacity basically represents the cell's ability to adapt to additional energetic demands in the cell. And so when you look at our actual data, what we found was that we saw quite significant differences when you compare our foamy macrophages. So our non-foamy macrophages are shown here in black and our foamy macrophages in blue. And I separated these out just for ease of comparison, but you can see here, this first reading, the basal respiration in the foamy cells is significantly higher than that of the non-foamy macrophages. Additionally, when you add oligomycin, you do get a larger decrease in this oxygen consumption rate, which indicates that there is greater oxygen consumption connected to ATP synthase activity. However, some preliminary studies that I've done in measuring actual ATP content actually indicates that the foamy macrophages have the same, if not less, actual ATP in their cells. So this makes sense when you look at the rest of these measurements, such as proton leak, shown here in purple, which again is that remaining oxygen consumption after you inject oligomycin. And you can see here it's significantly higher in the foamy macrophages, indicating a degree of mitochondrial uncoupling occurring. And lastly, when you inject FCCP, you can see a greatly decreased spare respiratory capacity in our foamy macrophages. Again, showing that these cells basically are acting right now at their theoretical limit, and they're basically, there's no additional room for them to respond to any stresses. So overall, this kind of paints a picture of the cell being in this extremely stressed state of, trying to meet this same energetic demand, but in the face of greater mitochondrial uncoupling, as evidenced by this increased proton leak and this decreased spare respiratory capacity. And so that made me kind of think more about this increase in basal respiration, basically what is accounting for this. Obviously, we have these indicators of mitochondrial uncoupling, including the proton leak and decreased spare capacity. But we also have this increased energetic demand in attempt to process these lipids. And so it's been well documented that, this can cause the production of reactive oxygen species. So that was kind of the next thing I wanted to look at as a result of these data. Oh, sorry, this is just basically the same data I just showed in the other, in this previous slide, but with each of the parameters individually expressed. So here again, we have our increased basal respiration, our decreased spare respiratory capacity in our foam cells, our increased proton leak, and the increase in ATP production. This also shows a decrease in coupling efficiency, which is kind of what I had attributed it to before. Coupling efficiency basically looks at both the ATP production as a function of basal respiration. So how effectively is your cell converting its basal respiration into ATP? And when you when you compare these two, you see that the coupling efficiency is reduced in the foamy macrophages. So as I mentioned, as a result of these indicators of mitochondrial uncoupling, but also this higher level of basal respiration, I also wanted to test the formation of reactive oxygen species, because as we know, mitochondrial uncoupling, when you're still in an energetic state, but and attempting to synthesize ATP to meet this increased demand, but you're also highly uncoupled, this can cause reactive oxygen species production. And so here we used Mitosox dye, which is a reactive oxygen species specific dye. And here we have our media-treated cells, and you get some baseline reactive oxygen species production, which is normal for some cells. The dye itself can also produce some instability, so that's also important to note. But when you add a inducer of oxidative stress, such as hydrogen peroxide, here you can see obviously a robust increase in the mitochondrial signal. And then similarly, when you look at our foamy macrophages, you also see an increased percentage of cells that are mitochondrial positive. And so this is just quantified here, mitochondrial positive cells as a percentage of total cells. So in conclusion, what we've seen here is that these foam cells exhibit certain parameters of mitochondrial coupling, including increased proton leak, decreased spare capacity, respiratory capacity, and decreased coupling efficiency. Additionally, we also found that one downstream consequence of this mitochondrial uncoupling is the increased production of reactive oxygen species. And so this kind of is a really interesting and exciting avenue for us to potentially look into some additional targets in relation to this pathway, because we haven't really, there's not much work done on targeting macrophage metabolism in spinal cord injury. So this could be definitely an interesting, avenue that we'd like to pursue in the future. So lastly, I also would like to share the work I've done in looking at the inflammatory profile of these cells. As I mentioned before, in the context of atherosclerosis as well as spinal cord injury, people have been highly interested in the role of macrophages, specifically lipid-laden macrophages, in inflammation after spinal cord injury. So we really wanted to get an unbiased glimpse at the inflammatory signature of these cells. So we used a cool system known as the Proteome profile cytokine array. And basically what this is it's a protein-based assay that you have 111 cytokines, antibodies, basically attached or adsorbed to the array membrane. And these are done in duplicate. And basically you can incubate these membranes with your cell lysate of interest. In our case was the macrophage and foamy macrophage. And basically, you can probe and visualize these, the degree of protein expression from these two membranes, and basically compare the inflammatory signature of these two cells. And here is just the cytokine array coordinates, which is how you can basically compare your two different membranes. And so we found 49 significantly altered hits, but I just highlighted some of the biggest changes, biggest fold changes that were evident in our non-foamy and foamy macrophages. So here you see an array membrane that was treated with lysate from just regular media-treated cells, and then those same cells treated with 1% homogenate for 24 hours. So when you compare these membranes, you can see an increase in a number of cytokines, including CCL6, IL-1 alpha was quite a big increase, as was tissue factor, or F3, shown here. CXCL16, also shown in green, was also highly increased, as well as GDF15 and SPP1. Interestingly, really the only statistically significant decrease in cytokines that we saw was MCSF. So that's in pink, you can see here was decreased in the foamy macrophages. So this is just these top hits in that the full difference between the two were the highest. So these are just taken from all four of the biological replicates that I used. You can see the media treated and homogenate treated dot plots from all 4 of those. So here's IL-1 alpha, SPP1, CCL6, tissue factor 3, GDF15, CXCL16. So when you combined all the statistically significant hits, not just the largest ones, we subjected these to, so here are the ones that I kind of highlighted in the previous slides, but there was also quite a few other ones that were significantly changed. So for these 49 cytokines that were significantly upregulated or significantly altered upon homogeneous treatment, we subjected these to a TAG pathway analysis, and James from our lab helped me do this. And some of the pathways that came up as being significantly enriched in this cytokine list was the age-range signaling caught my eye, because we have been, previous work of mine has been interested in the role of mTOR, which is known to be involved in this PI3K AKT signaling pathway. So as proof of concept, I actually had access to an mTOR inhibitor, Taurin, and basically treated my foam, either pre-treated with a vehicle or with Taurin 2, and then added homogenate in the presence of these inhibitors, and basically compared lipid accumulation in our untreated foam cells versus our Taurin-treated foam cells. We actually found a huge decrease in lipid accumulation in these cells, and very little cellular toxicity, which was we were very happy with. So something also interesting to note, I haven't really looked further into it yet, but definitely will in the future, was also a decrease in the actual distribution of the lipid droplets. You can kind of see here in the Taurin-treated cells that most of the lipid droplets are accumulating in the periphery of the cell. So the functional significance of this is still not quite known, but is also very interesting, and so something that we're definitely going to pursue further. So in conclusion, we now have our unbiased inflammatory profile of these cells, which includes a number of factors, including IL-1 alpha, tissue factor 3, SPP1, GDF15, CXCL16, CCL6. We performed a pathway analysis of all of these hits, all the statistically significant hits. We found a number of pathways that were changed, and specifically targeting this PI3K AKT pathway via mTOR inhibition resulted in decreased lipid accumulation in our foam cells. So kind of our take-home message, again, this is kind of my overall goal, was to really help understand more how these lipid-laden macrophages are contributing to the cellular pathology after spinal cord injury. And so I looked at the role of myelin after spinal cord injury and found that myelin is actually not the only nor the most important source of lipid debris after injury, and that we now have this modified in vitro assay that accounts for this and gives a more physiologically relevant way of testing these cells in vitro. Additionally, we started to characterize some of the metabolic changes that are occurring as a result of this lipid accumulation. And we found various markers of metabolic dysfunction and mitochondrial uncoupling. And this resulted in, or this was associated with increased reactive oxygen species production. And lastly, we also better understood the inflammatory contribution of these cells to the injury site, found a number of inflammatory cytokines that are upregulated upon homogeneous treatment. And we also looked at using these cytokines that were upregulated, looked at some potential pathways that were implicated in the development of foam cells, and successfully tested one, including the PI3K AKT pathway, and were able to successfully attenuate foam cell formation. And with that, I would like to thank you all for listening, and of course, thank my lab, specifically my mentor, Dr. Jay Lee, who's been great and so supportive throughout all this. And of course, my current lab members, Susanna, Lindsay, and James, who are also wonderful to work with and provide great collaborative environment. Of course, Yadira and Shafayat for helping run our lab basically, and making sure our animals are taken care of, and our previous grad student, Stephanie. And of course, my committee, Dr. Hackham, Barnbilla, Hassan, and Armando, the Marias Lab for their help with Seahorse machine, and then also Yanshi and the Fornoni Lab, and Hassan for assistance with opera imaging. And lastly, Adnan Mukhtiar and Constantine from the Park Lab for helping me image and form the cytokine array. And of course, all our funding sources. And yes, with that, I'll stop my share and look at the chat. Do you have any questions? So does Torin treatment affect endocytosis? This is a question from Nagi in the chat. And his question is, does Torin treatment affect endocytosis? If that is the case, you can assay endocytic markers. And to answer your question, it does. It definitely affects, from the reading I've done, it affects the lysosomal endosomal processing pathway. So I actually have a inclination that is partly the reason for the altered distribution of the lipid droplets that are now in the periphery of cell, and no longer kind of where you see a lot of the endocytic and lysosomal vesicles kind of in the center of the cell. So I'm going to be doing some lysotracker experiments, and I can also do some of the endosomal markers, as well as the autophagosome markers as well, to see which of these pathways are being altered. I don't know if there's any other questions. I think Marie can unmute your mic. I'll wait a couple minutes, and if not, thank you everyone again for.
Dr. Brambilla has a question, and
yeah,
her mic is
this is Berta. Very nice talk. I just wanted to ask you, did you have the chance to look at whether you have changes in lipid accumulation in the microglia, especially in your flow cytometry experiment? Can you look at body pH accumulation in microglia?
Yes, we did. So the microglia don't. They don't have. There is a population of microglia that does have the BODIPI signal. They're definitely not the majority, and all of the cells with the BODIPI high signal are like 95% macrophages. But something that was interesting was that in the MVP knockout mice, even though there's this BODIPI low population, there did seem to be a shift towards a slightly larger microglia population that was expressing this lower BODIPI signal. I guess that makes sense in context of obviously less macrophages being there. The microglia probably take on a compensatory role in assisting the phagocytosis. So I don't know exactly, it was kind of like another interesting turn that we could take, but we haven't really looked much further into that, but we did see a slightly increased BODIPI low population in the microglia.